and molecular structure of these proteins. And then in recent years, looking at their contributions to behavior, including autism. And today he's going to tell us about flies, I think, and how you take mutations that are related to autism and learn some interesting things by studying them in an invertebrate. And probably also you'll talk a little bit about mice too, I suspect. Welcome.
Thank you so much, but I cannot share the screen.
Pramila, can you make it for him to share?
You can try again now. Be able to share.
Let me see.
Looks good.
Do you see the PowerPoint presentation, or let me see.
Yes.
Yes.
Do you see the comments, or you see just the PowerPoint presentation?
Just the presentation.
Okay. All right. So, well, thank you so much to the Brain Foundation and Randy for the kind invitation. And our group has a really strong interest in understanding how disruption of neurocircuitry or molecular mechanism can lead to neuropsychiatric disorder. In particular, we're interested in understanding the role of dopamine dysfunction as a complication or participating as a mechanism in the etiology of amphetamine abuse, and for the purpose of this talk in autism spectrum disorder. So, for example, we are interested in understanding how amphetamine hijack particularly neurocircuitry in the brain in order to cause dopamine dysfunction and amphetamine abuse. We're also interested in understanding how single nucleotide polymorphism and de novo mutation in the dopamine transporter gene can disrupt dopamine transporter function, causing impairment in dopamine neurotransmission that we phenotype with specific behavior. And we use an array of tools to study these genetic variants, from structural biology to high-speed camera to look at the behavior of these flies. So dopamine dysregulation has been implicated in a number of neuropsychiatric disorders, ADHD, schizophrenia, neurological disorders such as Parkinson's disease, and lately also to autism spectrum disorder. In fact, some of the mutants that we analyzed in the past in collaboration with Randy Blakely has been found both in patients with attention deficit hyperactivity disorder and also in patients with ASD. So why ASD and dopamine dysfunction? There are a lot of traits that relate to autism spectrum disorder that points to dopamine dysfunction, from repetitive behavior, lack of rewards for social interaction, hyperactivity, anxiety, and so on. So in the scientific community in the last few years, starting to publish paper looking at the dopamine transporter dysfunction and his association with autism spectrum disorder. There are not many papers that have done that, but like I said, in the last few years, particularly our group, in collaboration with Randy too, has published a few papers regarding this subject. So just for giggles, there are two main dopaminergic projections. One is from the nigra to the striatum, and the other is from the ventrotegmental area to the nucleus accumbens and prefrontal cortex. What is important for this presentation is that the dopamine transporter is the main protein clearing the extracellular dopamine level once dopamine has been released. And really, it's been a renaissance for the dopamine transporter lately, because we discover a lot of very interesting mutations related to neurological disorders such as Parkinson's disease, of infantile early dystonia, and neuropsychiatric disorders such as bipolar disorder, ADHD, and ASD, that give us really the opportunity to apply all the knowledge that we have acquired in the last 10 years about the dopamine transporter to understand really how this mutation disrupt transporter function and leads to specific behavior. So this is the topology of the dopamine transporter, is 12 transmembrane domain, and up to now 5 mutants has been characterized in the dopamine transporter that has been associated with autism. they're all in critical structural domain of the transporter. One is the N-arginine for a tryptophan at position 51 in the N-terminus of the transporter. That is an important structural domain regulating transporter function. There is a deletion, an in-frame deletion of an asparagine at position 336 on the third intracellular loop. This is another mutation that is very, very important because the third intracellular loop is one of the loop regulating the intracellular gate of the transporter. Transporter have two gates, one extracellular, one intracellular, that are controlling and regulating the movement of dopamine across the plasma membrane. There is a alanine to a valine on the top of transmembrane 12. This was a mutation that was first discovered in a bipolar patient, later on at Vanderbilt and described by Randy and me for attention deficit hyperactivity disorder. And again, with the Vanderbilt patient in autism, again, characterized by Randy and myself. And finally, the first de novo mutation on the top of transmembrane 7, it is a threonine to a methionine. So when we look at the dopamine transporter, the physiological function of this protein is, how I said, is to clear extracellular dopamine for the extracellular space and move it inside. And typically, all this variation in pair transporter function, at least the one that we analyzed, by two different modalities. The first modality is simply that the transporter goes on the plasma membrane, but is not capable to reuptake dopamine anymore, right? And so there is an impaired dopamine uptake. And the second modality that is much more interesting for me, because I'm a biophysicist by training, is that now the transporter runs backwards, right? For reason that can be signaling pathway or structurally, now the transporter binds dopamine from the inside, the neuron, it pumps dopamine outside, increasing it for therefore extracellular dopamine level, and is what we call anomalous dopamine efflux. And this has been noticed for the first time in the A559V mutation, and later on in the T356M mutation associated with autism. So the proband is a male, has been classified as with autism spectrum disorder at age 5. And one of the important things about this threonine in position 356, it is highly conserved across phyla, from human to C. elegans. And the reason why it's so conserved, because he lies within the permeation pathway of dopamine and the co-transported ions, in this case, sodium. So you can imagine that any spatial constraint that the mutation threonine to a methionine can produce can impair, of course, the function of the transporter. And so like I said, we use a lot of structural tools from molecular dynamic simulation to structural biology, and also electron paramagnetic resonance spectroscopy to measure really how these mutations are impairing dopamine transporter function by looking on a structural perspective. So in collaboration with Samu Charab at Vanderbilt University, we do EPR, so electron paramagnetic resonance spectroscopy. So we engineer cysteine that are spin labeled, and EPR allow you to monitor the distance of these cysteines that we use it to look how the intracellular gate or the extracellular gate of the homologue, the bacterial homologue of the dopamine transporter is. And we look how these mutations are affecting the movement of the intracellular gate or the extracellular gate of this protein. And so without going into much details, what we saw that the intracellular gate was not really affected by the mutation, but we were looking at the spin label that we position on the extracellular gate of the transporter in the presence of sodium, the co-transported ions, and the substrate, in this case leucine, right? In the wild type, there was a rearrangement of the extracellular gate that was going in an occluded pore, right? So the transporter was trying now to move dopamine from the outside to the inside by closing the gate. But when we look at what was happening on the mutant, we saw a higher probability of these two cysteine, the two cysteine label, to be at higher distances, meaning that now the gate was opening to the outside. So when it was binding the substrate, moving the substrate outside the cell. So this was really the first structural fingerprint of what anomalous dopamine efflux was, meaning that the transporter, when it binds the substrate, spewed the substrate outside. So we had to choose a model now, an animal model, to start to screen all these mutations. And of course, mice cost a lot. And these we were running on fume on these projects, right? So we choose Drosophila to express the human dopamine transporter or the mutated dopamine transporter in the right neurocircuitry, meaning the dopaminergic system. So we use the US GAL4 binary system now to express, like I said, the human death or the mutated human death in dopaminergic neurons of flies that are lacking the Drosophila dopamine transport. And so the first thing that we've done, right, is to look at the fly brain that you see over here. There are a lot of dopaminergic projection. You see them in red, and to start to figure it out how to study anomalous dopamine efflux. Again, the transporter running backwards. Before the work was done only in cells, and now we wanted to move, of course, to an animal model. So what we use is amperometry, is a technique that take advantage of a carbon fiber that we position in any dopaminergic node of the fly brain. And this carbon fiber measure the level of dopamine by oxidation, right? It is represented as a positive current. So you can expect that if you have an anomalous dopamine efflux, the baseline recorded by this electrode will be higher than in the wild type. You will also expect that if you block the transporter, either with methylphenidate or other blocker like cocaine, this baseline goes down because you're blocking the constitutive dopamine efflux of dopamine, and this is what we notice over here, right? So you can see that you're already cocaine and the baseline goes down because you're blocking the constitutive dopamine efflux of dopamine. And we've done several studies characterizing structurally and the signaling pathway regulating the anomalous dopamine efflux, including we saw for the A5NV mutation, there was phosphorylation of the N-terminus of the dopamine transport. So what does it mean, this behavior? The first thing that we did is to convince ourselves that flies was a good model to study neuropsychiatric disorder. And we asked the help of a very famous scientist, Charles Darwin, and in one of his books, The Expression of the Emotion in Men and Animals, said that even insects express anger, terror, jealousy, and love by their stridulation. And so we thought that we were on the right track. And so the first thing that we looked, it was how the anomalous dopamine efflux was influencing locomotion, because a lot of patients with ASD are also hyperactive, right? And so I want to show you a movie. This is video tracking of flies expressing either the human death or the human death that now has been mutated. And you can see that the flies with have the human death causing anomalous dopamine efflux, they are moving much faster. They're crossing the center of the cage much more, and they're also they're grooming much more than the flies. They're just expressing the human death, and this is the path that the flies that are expressing the human death with the mutation are performing during this time. You can also do beam crossing to look at the circadian locomotor activity. So these are the wild types, so the human death over at the turn on of the lights and the turn off of the lights over 26 hours. And you can see clearly that the human death expressed in the T356M is completely hyperactive, almost similarly to flies that are not expressing the transporter at all, and therefore have a really high level of extracellular dopamine level. And you can quantify this over 24 hours to see clearly that the mutation causes hyperactivity in these flies. So another characteristic in autism that a lot of young people with autism spectrum disorder have a really high level of anxiety, with fear to be one of the most common subtypes of anxiety in autism. And so the question is really how to measure fears in flies, right? Animals, I'm a really outdoor person. An animal, I know that they fear under predatory pressure. And they fear to camouflage, but they fear also because they are scared, right? And the question was how to scare flies, right? And one time I was in my cabin where I pick a lot of mushrooms, right? And I saw one of these guys is a predatory wasp close to a nest of flies. They emit a sound like this. I don't know if you can hear it. And so the flies are scared. I'm sorry to turn off the sound because it was making me deaf. And the flies are scared simply because the predatory wasp is eating their larva. And so what we did, we recreate the sound of the predatory wasp in the lab. We use a high-speed camera. This camera can record 50,000 frames per second. This is a video that we recorded at 3,000 frames per second. And if you don't see here the sound of the predatory wasp, there is a red highlight of the cage that will tell you that the sound of the predatory wasp is coming on. And we're measuring freezing behavior in these flies, right? So you can see the flies move around. The video is being slowed down. The sound come on, they freeze, and they escape. Right? And this is a natural behavior. Usually animal freeze, and then they look in the environment, they escape. And so this is the mutant instead. And you can see that the freezing time of this of the mutant is much longer than the wild type. You can even notice it by eye. So the mutant moves around. It really doesn't move anymore. Right? So you can quantify this average velocity of the fly every millisecond, right? And this is the wild type in black. The animal moves around, the sound of the predatory wasp comes on, the animal freezes, and then is escaping. Instead, if you do the same type of experiment with the mutant, the animal moves around, the sound of the predatory wasp comes on, the animal freezes, and it keeps staying in a freezing state. Finally, we wanted to look at social interaction in autism that are impaired, looking again at what anomalous dopamine efflux does to this social interaction. So we have to find ways to measure social interaction again in flies. And there are several ways. I thought that this would be a cool way to look. You know, if you look at the school of fish or flocks of flies or birds, they maintain social distances. They maintain social distances because they try to survive in the environment, particularly when a predator is around. This is a school of fish that's being predated by a duck in the back. You can see it over here. And one thing that you notice is that the school of fish compress and expand depending on the distance of the duck, right? And so this is a behavior that is being modeled to say that when the animal, the predator approach the flock, the school of fish, the school of fish will compress and then will immediately expand because they try to escape altogether away from the predator. So we thought that we could do the same using the flies, meaning that we can measure the social distances of the fly, the cumulative social distances over time before and after the sound of the predatory wasp. And so this is what we saw. So this is the wild type, so expressing the human death, and it's at the on of the sound of the predatory wasp, they compress and then they expand. And if you look instead at the mutant, first of all, they start from a compressed state, right? But at the on of the sound of the predatory wasp, they expand and then they compress. So they do the opposite of what the wild type are doing. And so what we are interested in is not only about the human dopamine transporter, but also other proteins, right, that participate in the signaling pathway that are regulating dopamine transporter function. As we talk, we're working on syntaxin 1A. This is a protein that regulates the ability of the transporter to reuptake dopamine. Today, I show data that parallel a mutation, the first novel mutation in the dopamine transporter, that cause anomalous dopamine efflux. We characterize this mutation structurally, so we fingerprint what anomalous dopamine efflux occurs for this mutation, that is the opening of the extracellular gate, allowing the dopamine to be released outside. We also measure dopamine efflux, anomalous dopamine efflux straight in the brain of flies, and link this to impairment in specific behavior of the fly, including increased locomotion, increase fear response, impair social distances. Of course, the question remains, what is happening to more complex behavior that cannot be studied in flies? And so we have done a knock-in mouse, right? And we show in the knock-in mouse that we have impaired dopamine clearance, decreased tissue dopamine content, increased locomotion, impaired social behavior, and loss of social dominance. And you can see all the data published in a recent article in Journal of Clinical Investigation. I do have to say thank you to a lot of people, particularly Gabriella Di Carlo, Peter Hamilton, Aparna Shekar, Jenny Aguilar, and Christine Saunders, Andrea Belovich, And outside the lab, to Greta Galli, my daughter, that did some of this experiment, Kevin Errigan and Einar Mattis. Einar Mattis is an expert in genetics of flies. Mark Wallace, who together, the geneticist James Sutcliffe, and finally for the EPR to Assam Musharraf and Shruti Sharma, and for you all for listening to me. Thank you so much again.
Thanks, Aurelio. It's just amazing to see these conservation of these behaviors. Obviously, you know, a fly is not going to be a mouse, is not going to be a human, but we can trace these different systems, as you showed, you know, nicely with the fish and the flies, and the way in which we have protective behaviors ourselves together. It's just amazing. I have a couple of questions just of my own to remind me. And, you know, I'm sorry, I should remember all of this, but in the fly model that you introduced us to, was that the human mutation in the fly background, or was it in the human background?
It was, so we express the human dopamine transporter directly in flies in dopaminergic neuron. So these dopaminergic neurons are lacking the Drosophila dopamine transporter. So quote unquote, the flies get humanized with the human dopamine transporter.
I see. And do you put the wild type in parallel as your control?
Yes. So
okay.
Yes, and we backcross this fly quite a bit. So.
And does the animal with the human dopamine transporter look, and just the wild type, does he behave just like a wild type fly with a fly transporter?
You know, we have done this for locomotion, and they do, right? You know, the high-speed camera experiment are really complicated. So we don't have a parallel with the Drosophila dopamine transporter, but we always compare it to the one expressed in the human dopamine transporter. In terms of uptake and efflux, they are the same. So the one expressed in the human dopamine transporter are really similar in terms of clearance and response to psychostimulants, for example, to amphetamine, to what we have seen with the flies expressed in the human dopamine transporter.
So maybe with time, because these other domains that are probably divergent have regulatory elements in them, and you might find some really interesting things where the transporter is regulated, maybe not appropriately because of the sequence divergence.
Yeah, we are working heavily now on the microbiome to look how it's influencing transporter activity. Right. So.
Right. Yes, Dr. Galli has quite a nice program going on microbiome, and with his dopamine background as well. If anybody's interested in asking about that either now or later, I'm sure he'd be interested in hearing your questions. I have one more question about flies and how you use them. So a lot of people will turn to Drosophila because they can screen for mutants or screen efficiently, genetic stocks that have mutations in a whole bunch of things. Have you explored yet any of these genetic variants that might, if you put them together I think that your T356 mutation, this de novo mutation from individuals with autism, is a very powerful reminder. I mean, it's powerful evidence that the dopamine system can contribute to autism, whereas the rest are probably more risk factors and need other things to come together. So that was what sort of made me think a bit about some of these other mutations that might be interesting to put together. Have you put any together with yours?
No, but the T356M is actually found now in another patient in Cincinnati, and we're in contact with the doctor. They just swap the saliva and send it to us to look at the microbiome, and also in a new patient in Europe. So it's becoming a very interesting mutation to follow up in all these kind of things. In the mouse, for example, the one that I show in the knocking mouse has a difference in the fusobacteria in the gut, right? It has been linked to dopamine dysfunction and in autism. And we measure the microbiome in this mouse, and now we're trying to compare this with the patient.
Dr. Blakely, there is a question in the Q&A.
Okay, let's see. Can I find it here?
Can
Okay, what do we know about neuromotor issues in the mutants? How do we go from that to behavior response? Aurelio, you showed us hyperactivity. You also showed us repetitive grooming behavior. Any other motor responses that might interest this audience in relation to?
So in the knocking mouse, they are performing much better on the rotor rod. That is another motor response that I remember now on the top of my mind. And the mouse is hyperactive too. I try to remember other things. In the social dominance test, they lose, like you show for your mutant and the serotonin transporter. That is about it that I remember that we've done. And the mouse is available to everybody. So whoever is interested in using this mouse, just email me, and I'm more than happy to send it to you.
So maybe, Ashok, if you have a more refined question about neuromotor issues, were you thinking about something in particular? But Ashok has another question about that microbiome. He wonders about specific metabolites that might be secreted that could be responsible, I think, for some of the behavioral changes that you're seeing.
What other metabolites do you have in mind?
Are there bacterial produced metabolites that might be affecting brain function?
Okay, so we didn't look at that. So the fusobacteria is producing butyrate, and we have a model in actually amphetamine abuse, because a lot of methamphetamine abuser has increased fusobacteria in the mouth. That is why they have gingivitis. And so when I was reading and finishing up this paper, hopefully soon, on butyrate and amphetamine and methamphetamine abuser, I discovered that in autism, it looks like this kid has low level of fusobacteria. So we got a knocking mouse, and this was the first thing that we've done, is to look at the fusobacteria in this knocking mouse, and they are low. We didn't measure butyrate because it's actually quite difficult to measure. Right?
Well, that's going to be interesting to follow up. One of the things on the serotonin side, serotonin obviously in the gut affects the microbiome as well. And we've been trying to get our mutation in a cell-specific knock-in, which is a little harder to do than the transgene or just a standard knock-in to do it conditionally. But I have wondered how much of the behavioral phenotype that we have is due to what's happening in the gut being transmitted to the brain, just like the immune function, right?
Yeah, in flies, butyrate influence a lot of this behavior. It's influencing courting behavior, is influencing locomotion. So we feed the butyrate directly to the fly, and we modify the behavior of the fly. Now, we didn't do it in the mouse.
It's an interesting story about amphetamine, and I understand why you would do that, given dad is the site of mutation. Let's see. I think that's the end of the questions that I see. I'll thank you again, Aurelio, for
thank you so much.
A very interesting talk and providing this insight, this evolutionary insight into how we study autism mutations. And now move to our next speaker. So, of pediatrics at Rutgers, New Jersey Medical School, associated with St. Peter's University Hospital. And her work has looked at neuroimmune interactions and epigenetic regulation in children with autism. She also has interest in immune abnormalities and peripheral disorders such as asthma and GI dysfunction. So it's a pleasure to have you join our panel, and I'll turn it over to you now.
Did you see the slide?
No, please open the slide and then hit share screen and then select.
Okay, share screen. Okay.
Yeah, and select the presentation.
Yeah. Oh, I hit the share screen.
Does it show you the presentation in the panel?
Yeah.
Just select that.
Let's see. You don't see that? Let's
Not
see.
yet.
Ooh, okay. I think that I hit the so that I hit the screen. Did you see?
Yeah.
Now we do.
Okay.
Now we're getting there. Yeah. And Harumi, you might want to turn your camera down just a little bit because we can only see you from the nose up. Perfect.
Okay. Yeah.
Perfect.
Sorry. Yeah.
And if you can please go to the slideshow. Yeah.
Okay.
Thank you.
Yeah. Now it's okay.
It's good.
Yes.
Okay. Yeah. And thank you for inviting me, and also very generously funding this project. So this is a progress report of the project that funded by the Brain Foundation, and essentially focused for the innate immune memory biomarkers as a part of the neuroinflammation. So, sorry. So the Brain Foundation funding this project has two aims. Number one, I have been working on the monocyte cytokine profiles as a marker of the innate immune response. And we want to associate with the clinical characteristic of those cytokine profiles. And the reason why we looked for the monocyte cytokine profiles, that's as a marker of the innate immunity. Obviously, innate immunity has been thought about for the some implications of neuroinflammation in other type of the neuropsychiatric conditions, and it has been worked as for the in other common neuropsychiatric conditions such as depression, schizophrenia, something like that. Then why the innate immunity and the innate immune memory was really implicated? All these neuropsych conditions, all the studies tried to implicate with one specific pathogens associated with condition has been all failed. So all the conclusion is really non-specific immune response, which is typically the innate immune responses associated with onset and progress of the neuropsychiatric symptoms. But then its question rises, why is the innate immunity, which doesn't have any memory, but a lot of condition once activated, then it's very lasting effect. So obviously the innate immunity doesn't have any memory for specific pathogen, just like if we have a measles, we have a very prolonged memory for measles. But then, about like 10 years ago, from that Dutch group, there is a concept of the innate immune memory, and which is essentially the epigenetic changes of the monocyte or macrophage populations. And those monocyte or macrophage and also other innate immune cells are so true. They're all later found out there is an innate immune memory generated in response to fairly strong immune stimuli. And those changes were initially metabolic changes. And it's usually ATP, ATP production to more getting into the glyconeogenesis, then accumulations accumulate, which is changing to a histone modification, affecting the histone acetylase and histone methyltransferase changes. So essentially a lot of things associated with a mitochondrial functional change. Then also it's a prolonged case, it has been shown there is a change of the histone modification, it's I'm sorry, it says I misspelled. Histone modification markers is changed. And this is for the ASD. And so that's a part of the reason we check first monocyte cytokine profile, and then how associated with clinical feature. Then how these condition was also any associations, monocyte histone modification markers. And then in the selected patient, we're going to do the super enhancer chip sequencing. Then which target to be used for super enhancer chip sequencing, we will decide based on the expression of the histone modification markers. Then M2 is for the more likely the quick assessment of the serum markers, which can be used for screening of those changes. Then previously we have done this deep sequencing of the microRNA in serum. And the reasoning why we did that, because the major source of the microRNA in the serum is a platelet and monocyte macrophage. And also the microRNA as secreted as a form of the, it's like a cyst with like a, it's fairly stable and secreted as like increased in the endosome. So that's the reason why the microRNA, because we did a long time ago, we did the serum cytokine levels, and those are not very helpful. So we wanted for the little more reliable, like a serum marker. So these are two purposes of the our proposed study that the Brain Foundation funded. And then we tried to do the this, we haven't completely finished, but so that it's some of the progress report. Some of AM2 is almost finished. So, okay. So the initial study for the associations between clinical features and monocyte cytokine profiles. So that was, we did that because the previous studies and also the more newly recruited. So we have done that about like close to 150 in ASD subject. And some of the clinical association, it was significant, is GI symptoms and TNF-alpha production. Then also for the IL-6 and IL-1 beta, that was more associated or fluctuating behavioral symptom. It's categorized as PANS, but I don't think the ASD patient has a real PANS. It's more likely the post-infectious neuroinflammation, and it does show the symptoms like anxiety, OCD, anxiety, irritability, and worsening hyperactivity type. And seizure disorders, which is not surprising, at least our data showing some association of the IL-1 beta production by monocyte. And we just made sure those associations are not associated with the medications they are currently on, because recruited patient is not really ruled out with the use of the medication. And also it wasn't affected by age, gender, and the other core variables we checked. And also we didn't see the significant association with the autism severity by ADOS. So then for we have done the histone modification marker expression. We assayed in the 48 subjects, and now we kind of in the process of another 24 subject. So this is for the preliminary, this is only preliminary result. Then addition of the data may change. But for the IL-10 production, by monocyte under the LPS, zymosan, or beta-glucan. So those are positively associated with expression of the H3K23AC, which is a typical like a histone activation marker. It's reported to be upregulated in the trained immunity, which is more likely the little more hyperactive innate immune memory. But usually for the H3K27AC is only temporary. Then other type of the intermediate, they tend to have the downregulated. But at least for the current condition, H3K23AC tend to be continue to be upregulated in ASD patient, which is a little different from the reported histone modification marker pattern reported in the trained immunity, at least mouse model and some of the human studies. So seems a little different from the ASD patient. And we also see that for and it's we did see the IL-10 production is a suppressor cytokine. So we did see we expected H3K23 as AC expression. IL-10 production supposed to be downregulated, so it's going to be a negative association. However, and that was true, at least for preliminary data for the non-ASD controls. But again, ASD subject has a more positive, like, it's not clearly positive. Some of them is, so it's not very, its association seems different. Yeah. But they did see the sum. So at least for preliminary data, those markers for the epigenetic regulation for trained immunity seems like different in ASD patient as compared to non-ASD control. So this is not so many patients because still in the very preliminary, but non-ASD patients appear to have some negative association between the IL-10 production and the H3K23AC expression. It's essentially the higher expression, it's a lower IL-10 production. But it's more like a positive association we see between the H3K23AC expression by monocyte and then for the monocyte IL-10 production. So that was also a little puzzled us. So we want to see further. Then at least we have looked for the 4 histone modification markers as markers of the histone trained immunity as well as the tolerance type. And the best significant changes were seen in the H3K27AC expression. So therefore, we have now doing the super enhancer chip sequencing, and the target molecule we selected as the H3K23AC. And then this assay is very costly, so that we only checking for the very significant patient of the various influence. So there are 2 different relatively stable ASD subjects, and it's more like a very lot of inflammatory condition and some non-ASD control. And it depends on the result, we try to scale down the little more less expensive, like a little simplified chip sequencing. Then we can do more samples. So, okay. So then this is like a progress report of the AM1. Then the progress report for the second part, AM2, this is we have more data, so a little more detailed result I can show. And this is as I explained, we wanted to have a good, very stable serum marker, and also very easy to be detected by almost everybody. So essentially, we selected from the previous microRNA deep sequencing result. We selected 7 microRNAs. Then we just checked it with a regular quantitative, essentially QRT PCR. Then it's like an expression of the result of the PCR count. It's a normalized PCR count. And I like, and actually now many people now using the normalized PCR count because it's a little small amount for present. These, the way that this is expressed, at least my understanding, is getting popular to express like this way. And also I'm familiar with this, the way that it's expressed because these, the way that a PCR count has been used for the newborn screening for the skit. So I'm involved in the newborn screening skit in the New Jersey. So I'm used to be the this kind of the way expression of the QRT PCR result. And so the essentially we used for 130 ASD subjects and 50 non-ASD controls. And so the essentially for the we didn't find so the significant changes from controls of these 7 microRNA PCR count when we combined all the ASD subjects. However, when we checked for the changes based on the comorbid condition, there is some changes, and those changes are most significantly observed ASD subject with or without seizure or sleep disorders. Essentially, the ASD subject without seizure or sleep disorders have a higher circulating level, which means lower PCR count as compared to the seizure ASD subject with seizure or sleep disorders or non-ASD controls. So this is for the result changes not that like a significant striking, still like a piece. This is Mann-Whitney test result, and there is for the like a seizure negative, there is some MI 423, and also for the sleep positive or sleep negative subset, there is a difference. And it's in general, PCR count is lower in the sleep negative, which means all these the levels of the circulating mRNA is higher. And so, and this is like a 0. Other 3 didn't have shown that the p-value, but it's very close, like a p equals 0.06 or 0.7. So it's a tendency to have this. So this is for the condition, and we did see the some like production and comorbid condition. So is there any problem? So we wanted to see if it was these microRNA PCR count, is there any association with monocyte cytokine production? And especially we thought if this is see some difference, maybe the ASD subject with or without seizure and sleep disorder maybe shows a different conditions, different association. And actually that was true. We did see some positive associations with production monocyte cytokines. It's a TNF-alpha, IL-6, IL-1 beta, IL-10. So these are kind of the cytokines almost. So that's why I was interested for the serotonin metabolism too. And so this was actually this kind of association is only more evident with without seizure or sleep disorders. The patient who has a seizure or sleep disorders, they didn't have a good association. Essentially, microRNA levels stays low, then cytokine production stays up. That is a type of the patient who has a lot of sleep disorders and seizures. And interestingly, we didn't see such associations when in non-ASD controls. So this is for the how it's like associated with. So I didn't see the data for the okay. So I did see the some of the association or ASD total, and this is we just showing the spontaneous TNF-alpha production, because this is probably most likely reflecting the in vivo status. Then we didn't show any data with ASD with seizures or with sleep disorder, because we didn't see any significant association. And the result was the p-values shown is a result of the Spearman test, but it's essentially similar result we also obtained with Kendall-Taubes test as well. So essentially pretty good association, especially when we see this, when we looked into this association with ASD and without sleep disorders. And this is a TNF-alpha production, and the same kind of the association we see IL-6 production. But it's not as TNF-alpha production, we did see the more good association. And the same way for the IL-1 beta production. But IL-1 beta production, we did see some association non-ASD controls. But this is again opposite. This is for the no positive association for the PCR count. It's actually the negative association. So this is again quite a different association that we see with those microRNA, circulatory microRNA levels and the cytokine production. And it's also, again, we did see the spontaneous IL-10 production and miRNA levels. Again, this is for the sleep ASD patient without sleep disorder or without seizure. It's a good positive association. Okay, so the essentially for how if the serum circulatory levels of miRNAs are associated with those cytokine production, then also we saw that some GI, some comorbid conditions, we saw that previously some associations with monocyte production. So we want to see if those kind of if circulating level of these 7 microRNAs checked, does it change with some of the ASD subject? And this part we haven't completed yet, but at least in 3 patients in whom we are able to get the serum sampled repeatedly, and partly because they are relatively closely followed in our clinic and they are sick, so that we are able to get the serum samples. So this is for the patient. Case 1 is really very severe seizure disorders, and we have to use a lot of immunomodulating agent. We used the mTOR inhibitor and also anakinra. iron beta blockers. And he was also on the IVIG. And all the levels got better with seizure is actually video EEG completely normal. But after the hit by microbial infection, unfortunately, all the levels went up. And case 2 has been diagnosed with more like encephalopathy, and he has been giving a lot of mitochondrial rescue medicine like leucovorin high dose and IVIG, and also tried on rituximab, and a lot of medication tried on the autoimmune encephalitis. But his level is stays fairly high levels and never been getting the, PCR values high, means circulatory miRNA levels stays very low. And number 3, these are patients. The third patient is a GI, significant GI symptoms. But he did respond to a lot of probiotics and also have antifungal or other type of the dysbiosis treatment and dietary intervention. And in his case, his clinical features get better, means less hyperactivity, less anxious, and it's more like a more focusing without using any other medication. And it's nicely those circulatory, these 3 circulatory RNAs, microRNAs, PCR count is decreased, means levels are getting higher. So I think that those circulatory microRNA levels might be the good marker for the disease activity, and especially these levels PCR count high, which means circulatory levels stays low. There will be something problematic or something else we need to look into that. Yeah, and especially I was fascinated with sleep. Patient who has a lot of sleep disturbance have the very low levels. So those association, we need to look into that. So these are not, these circulatory microRNA levels are not a good like marker for autism diagnosis, but it may be a marker for neuroinflammation. And also we checked for what kind of the function these microRNA does. And we haven't completely finished, but it's mostly reported in the cancer study because they are like a suppressor of the cancer. So suppressor cancer means immunologically essentially suppress the immune activation. So those these microRNA is essentially the suppressor of the inflammation. But the number of the articles is much less as compared to cancer research. So we may have to do a little more functional assays. So essentially, this is a summary of the how and the essentially for the result of the circulatory microRNAs, and the low circulatory microRNA, no association with monocyte cytokine. Maybe we can't completely say that, but our data showing that low the microRNA means more like a stays higher monocyte cytokine profile. So I thought there might be something else is and it's retaining for the this high monocyte cytokine profile. Okay. So this is the progress report of the project funded by Brain Foundation. Then also this one is also funded by John T. Foundation. And the Brain Foundation funding is very helpful because the DHS, Department of Health and Human Resources of the New Jersey, they didn't fund us. There is no funding available because of the pandemic. They started funding a little bit this year, so we get a small amount. But we were able to continue this project because of the Brain Foundation funding. And thank you very much for that funding. Okay. And also, I didn't say, but thank you for all the clinical staff and also my laboratory, my technicians and all the collaborators. So that is my report. So I finish. I think
One question, if you want to answer that, and we can conclude. Thank you so much. It was a great presentation. Thank you, Dr. Blakely.
Yes, we have one question. Does dextromethasone or prednisone alter the monocyte marker profile? And is there any benefit to ASD patients?
I think because of the steroid changes almost everything. So with these assays, we avoided for any of the patient, within 2 weeks of the steroid use, because almost non-specifically all these cytokine production we know it's going to be suppressed by steroid. So.
And I have one quick question. Just that longitudinal profile that you showed, you had, I think, 3 points on it. And I didn't know when that started and how wide those.
Sorry. Yeah, because this is only for the case presentation. The starting point is the time when the patient enters the study. And for the seizure patient, that's before I started treating for the seizures. And then it's like a closely monitored. And so
what's the distance between your points?
It's usually 6 months to 6 months, 3 to 6 months. But each patient has a little different. So I didn't really do that very clearly. But for because we don't really protocol per se, we're supposed to get to the additional sample for 6 months. So we just followed it. But seizure patient have the striking difference, so that we get it a little early time point. Yeah.
I understand. Thank you.
So thank you so much. And thank you all for attending till the end, and see you all bright and early tomorrow morning at 9 o'clock. Thank you.
Thanks, Pramila.
Thank you.
Yes, thank you. Yeah.
Thank you.